Hi, I'm Sydney, and I'm going to be talking about Bayesian modelling of whole brain cell count data. Cell count data can now be collected across the whole animal brain, providing neuroscientists with alternative methods for capturing recent neural activity. Typically with these data, the number of brain regions recorded from is often much larger than the number of replications across animals. There can also be experimental factors that affect the data collection process, resulting in damaged tissue, or in some cases, the lack of an observation entirely. Together, these form a challenge for traditional frequentist approaches to analyzing the data, as they require large sample sizes for statistical inference. So why is the Bayesian approach a good one? Bayesian models can capture the structure of the experiment. They are generative models that can capture the characteristics of the data at its various levels. We can include relevant prior information in Bayesian models. Imputations also work well in the Bayesian framework. Imputations are estimates of missing data that provide inference with more information than it otherwise would have had if we simply dropped all rows from our dataset that contained a missing value. So a good model for these kinds of data should aim to capture the following. Theta. This is a mean cell count per brain region, or a mean expression level per brain region. We expect animals to respond uniquely to treatments, and this is captured as an offset about theta. Here it is written as gamma. And tau captures animal heterogeneity. It's a quantification of the diversity of responses of animals in each group and each brain region. So here is the model. I've colored the likelihood at the bottom in purple. As observations are count data, we've used a negative binomial here. Moving one step above this in the hierarchy, in orange, are the parameters that define the mean of the likelihood. Notice how gamma has been given a multivariate prior. This is because we believe that the unique responses across brain regions for each animal should not be independent of one another. Finally, the step above this in red are the hyperparameters. To demonstrate the performance of this hierarchical model, we applied it to some experimental data that examined the role of the nucleus reunions in the recognition memory circuit. This data contained 40 male rats. 23 brain regions were recorded from. The rats were required to perform a familiarity discrimination task, and this results in a 2x2 experimental design, leading to 4 groups. One immediate effect of using this hierarchical model can be shown with a comparison to a simpler model on a typical analysis for these data that looks at the effect of surgery through a difference in group means. On the x-axis are the brain regions, and on the y-axis are the difference in cell counts. The top line shows the difference between lesion familiar and sham familiar groups. The bottom line shows the difference between lesion novel and sham novel groups. Notice how in each case, the hierarchical model has much tighter posterior intervals than the simpler model. It's worth noting that the simpler model attempts to fit the same number of parameters as the hierarchical one. However, it drops any of the structure present, for example, the correlation matrix and the animal heterogeneity. This is important because take, for example, the ventral subiculum. For the simpler model, there is large overlap with 0, and it is hard to say if a difference is present or not. However, the shrinkage provided by the hierarchical model is much more supportive of a difference. To demonstrate the imputation performance of the model, observations were uniformly removed from the dataset, and true values were compared to their posterior estimates. These have been plotted here. Y-axis are the missing values, and on the x-axis are the true values. Overall, there is good overlap with the identity line of the posterior intervals, showing good recovery in almost every case. The separate graphs are for separate brain regions. As we fitted the model in Hamiltonian Monte Carlo, there can be difficulties with imputing discrete data, simply because Hamiltonian Monte Carlo is defined for continuous spaces. So for imputation here, we used a normal approximation to the negative binomial distribution. As mentioned earlier, the model contains a correlation matrix. Here, the Bayesian estimate of the correlation matrix compared to the sample correlation matrix have been plotted. The Bayesian estimate is in the lower triangular matrix, and the sample one is in the upper triangular matrix. Rows and columns have been colored according to a coarse grouping of brain regions. Starting from the top row, these are hippocampal regions, prefrontal regions, rhinal regions, and the two regions taken as control regions. These are the motor cortex and visual cortex. What's nice about this is that we can clearly see similar structure between the two estimates. Examples of these are given by the green squares. However, the Bayesian estimate is much more conservative than the sample estimate, which has many correlations larger than 0.8. These are probably unreasonably large. Notice that the Bayesian estimate is more stable as it loses much of the variation present in the sample estimate. In summary, Bayesian models are suitable for cell count data as they can capture the structure at multiple levels. They provide ways of dealing with missing data through imputation, and this is jointly at model fit. There is no separate imputation step. Information sharing in the hierarchical model provides shrinkage. This gives a reduction in posterior uncertainty and allows us to draw stronger conclusions of the model. Thanks for listening, and here are the references.